So dear students, today I am going to give the explanation of mechanism of crossing over. So this is one of the slides presentation prepared by student Tanjil. So as you know, crossing over, it's exchange of genetic material between non-sister chromatids of homologous chromosomes during meiosis. It occurs in prophase 1 of meiosis, that is Peckitin stage. So you can see the structure or the stages of prophase 1, because first of all you should know the different phases of prophase 1, how it undergoes, what changes are going in the stages of prophase 1 during meiosis, and then you come to know that in the Peckitin stage this process, the crossing over, takes place. So first stage you can see here, leptotin. In leptotin stage, chromosomes condense and become visible. They become more visible. Then in zygotin stage, homologous chromosomes, they form the synapses or they form the pair. Then in Peckitin stage, generally this is a stage when crossing over occurs. And in diplotin stage, chiasmata formation becomes visible. And in dikinesis, chromosomes prepare for the next stage. So this is how the stages of crossing over takes place during the prophase 1. These are diagrammatically shown in these pics. You can see leptotin, zygotin, Peckitin, diplotin and diakinesis. So now see, let us understand what is the process of mechanism of crossing over. So generally there are four stages. First is synapsis, second is formation of synaptonymal complex, third is breakage, and fourth is exchange. So synapsis occurs in homologous chromosomes as they align close together, they align closely. And then in the formation of synaptonymal complex, a protein structure that holds the homologs together, that is formed. And in breakage, DNA of non-sister chromatids breaks at identical points. And exchange, then in the fourth stage, exchange of broken segments are exchanged between the chromatids. You can see now the synapses, how the synapse is formed. This is the first stage. During meiosis, you can see these chromosomes. This one, it's shown by color blue and color red. And you can see both color chromosomes, they are forming the synapse. So it occurs during zygotin stage of prophase 1. Here homologous maternal as well as paternal chromosomes pair closely and the pairing process is called the synapsis. And in this way, in the center there is a complex formed which is named as the synaptonymal complex. So DNA molecules of homologous chromosomes, they exchange the segments. So function of synapsis is just provides a mechanical basis for heredity and variation. So this is the synaptonymal complex, how the synaptonymal complex is formed. You can see here in between the two chromatids, you can see crossover in the center, crossover is formed, and SC means synaptical complex is formed. And in synaptonymal complex, you can see red colored above loops, these are lateral element. Then lower side blue colored loops, these are again lateral elements, and in the center purple color, this is a center element. So synaptonymal complex consisting of lateral elements and center elements, and this is chromatin. So two lateral elements and one center elements. Then transverse filaments, they connect them. So these are the transverse filaments which are connecting. So synaptonymal complex is a three-part, you can say tripartite protein structure that is formed between the homologous chromosomes during prophase 1. So main function is it stabilizes a pairing and may help in recombination so that they can join together for some time and they can exchange the segments. Then next is the duplication of chromosomes. So you can see here in the structure, this one for yellow colored and orange color, brown colored, one segment and second segment. Then 4 chromatid stays. Then you can see here synapse is formed. Then you can see after the synapse formation, after separation, there are the one segment, one chromatid is yellow and another chromatid is yellow, half orange, half yellow and one orange or brown, last one is brown. So this is how they have exchanged the segments in between the two chromosomes. So it follows the synapses. Each homologous chromosomes duplicate into sister chromatids, forming a tetrad. So this is a 4 chromatid stage which is named as a tetrad. So these bivalent split longitudinally into 2 chromatids and attached by upslipped centromeres. You can see in the diagram here. Then crossing over occurs. Here it is beautifully explained with the help of colored chromosomes. You can see this one red color and blue color. Then how the after replication this one is chromosomes are formed. Homologous chromosomes undergo synapses, red colored and blue color. And after forming the synapses, we are having the different kinds of gametes in the form of when they separate. So you can see red colored, then one is fully red colored, another is red colored and slight blue color, one is blue color and slight red color, and other part of it is blue, completely blue color. So it occurs during pectitin stage. Non-sister chromatids exchange because these are not the parts of the same chromosome. These chromatids are the parts of the two different homologous chromosomes, and enzyme that causes a cutting or that acts as a scissor, that is named as the endonuclease enzyme. An enzyme which helps in the joining of these segments, that is the ligase enzyme. So crossing over may occur at several points, but generally it occurs in 1 or 2 points. So genes close together, there is less chance of crossing over. So you should remember the name of enzymes that cuts, that is named as endonuclease, and the name of enzyme that helps in joining the segment, that is a ligase. Then lastly, terminalization occurs, and in terminalization, after crossing over, chromatids repel each other except at chiasmata, and repulsion starts from the centromere, and chiasmata move towards chromosomes and this process of terminalization. So this is a terminalization of crossing over, and we have getting the new combinations in the chromosomes. So these having the both characters of male as well as a female. So it ensures a proper separation of homologous chromosome. This is how the terminalization occurs. So this is all about the process of or mechanism of crossing over, because in one sentence you can say that it's a process of exchange of genetic material between non-sister chromatids of homologous chromosomes during meiosis. But when you give the mechanism, it's having the different parts. First is synapsis, then formation of synaptonormal complex, then duplication of chromosomes, then crossing over, chiasmata formation, and terminalization. So complete lengthy questions about the crossing overs can come, and you can attempt in the length fully mechanism of crossing over. And some short answer type questions can also occur from this part, as define crossing over, or in which stage crossing over occurs, and name the enzyme which helps in cutting of the segments, and name the enzyme which helps in joining of the segments, and what do you mean by synaptonormal complex, and draw the diagram of synaptonormal complex. So these are some questions, expected questions you can prepare through the books. So go through the books in detail. So thank you.